Namaskar. Welcome to our channel Bio by Gulab Sir for Wisdom Coaching Center. I am happy to welcome you as a part of series of lectures based on 12th class syllabus, CBSE as well as state board syllabus. It is common for all the boards. This is the beginning of video on 12th class syllabus. First chapter is Reproduction in Flowering Plant. I am very happy to introduce you that our system is based on conceptual education, and you would have heard a very recent news that from this year, either November onward or next academic year onward, there will be a open book exam. You know, student, really it would be very interesting to the student, those who really study, understand through conceptual education. Simply mugging up and writing that answers in examination would not be rewarded. That questions will be critical, analytical and thinking based. Unless and until you have clear-cut understanding to the concept, you won't be able to answer the questions. So better to have conceptual study. It is very important. And yes, real study is that one who understands and write in his own word. That is always appreciated, and that is always with one who study through conceptual education. One who mug up may forget or may not explain the way it would have asked the way. Question can be changed. It is not so that what question given in books as it is will be asked. So question may be changed. In that situation, one who had conceptually studied, very clear-cut idea, understood very well, can understand the question and can apply the knowledge. So our education system, that wisdom education system, right from the beginning has given weightage for the knowledge and not for the marks. So marks can come behind knowledge, but knowledge may not come behind marks which gain by cramming or mugging up. So one who has that knowledge has capacity to explain, has capacity to understand the question, and therefore we have very clear-cut mission and vision that to make your concept clear. In our video, you will not be miss a single minute. That every sentence will have that some either of importance as a concept. So I welcome you. Although there is a new system and will be new system, that open book system, but these videos will help you to understand any kind of question, even if very critical. Let it be critical because concept is clear, you will understand and you will apply the knowledge the way we did learn through these videos. So let us start first chapter in 12th class, Reproduction in Flowering Plant, with the very first topic, development of male gametophyte. Before that, as a part of introduction of this chapter, as you know that male gametophyte is pollen grain. So in all sexually reproducing organism, that they have male and female gametes. So as plant, they have male and female reproductive organs. You learn in 9th and 10th class that male reproductive organs are stamen and female reproductive organs are carpel. Stamens made up of filament and anther, carpel made up of stigma, style, ovary. That is basic knowledge you have. So directly we have to start this from development of male gametophyte. This includes microsporogenesis and microgametogenesis. So let us begin. So here, as you are aware about the structure of this is stamen with anther and filament. This can be monothecous or dithecous, that you learn. Having two theca, dithecous, having one theca, monothecous. What this structure as this is anther and this structure is filament. This anther, this part, what we call theca, is also known as microsporangium. sporangium, because it consists of microspore mother cell. No doubt we are going to learn first about anatomy of this microsporangium and then development of male gametophyte. So what this microsporangium is? This microsporangium has centrally placed this sporogenous tissue, this centrally placed sporogenous tissue, also can be called as microspore mother cell, can be called as microspore mother cell. So each cell is these are microspore mother cells and can be called as sporogenous tissue. So to have the best understanding, you can refer that book simultaneously. That innermost layer which cover microsporangium is a sporogenous tissue, is covered by a nursing tissue, is covered by a nursing tissue. This is a single layer of nursing tissue called tapetum. So here this layer is tapetum. Now these tapetum has they have either 2 nuclei or 3 nuclei, because these cells has dense cytoplasm and has ability to undergo karyokinesis but do not undergo cytokinesis, and therefore it can be 2 or 3 nucleated cells. It is a nursing tissue that provides nourishment to the developing pollen grain during the process of microsporogenesis and microgametogenesis. So this layer is called tapetum. So here these are called microspore mother cell, microspore mother cell. This is tapetum, is a nursing tissue, can be called as nursing tissue. Next layer, these are 2 layers called middle layer. These 2 layers outside the tapetum called middle layer. Out of these 4 layers, these middle layers are 2, otherwise rest of the other layers are single. Outside the middle layer, this is endothecium. This is endothecium. See, middle layer is of 2 layers. Endothecium is single layer, and outside to this endothecium there is a single layer that is called epithelium. It is a protective layer. Including this protective layer, epithelium, these endothecium and middle layer help in dehiscence of pollen grain. So let us label after endothecium. This is epithelium. Now see, in outermost epithelium, second layer endothelium, two layered middle layer and innermost is tapetum. This is a complete structure of microsporangium, transverse section of microsporangium. Question can be asked for 3 marks. If it is open book exam, question will be asked that a reproductive organ which has 4 protective layer, outside layer, with including one nursing tissue, having tissue to develop the pollen grain. Draw a diagram and explain. So if you are understanding, definitely you will answer the question. You will draw. Question can be asked, a organ or anatomical organ. Explain the structure of anatomical organ where that carrier of male gametophyte is developed. Who is carrier of male gametophyte? Pollen grain. So question can be asked in critical way, different way, so that you have to have touch of that knowledge, then only you will understand. So this is an anatomical structure of microsporangium where these microspore mother cell undergo microsporogenesis to develop microspores, and these microspores later undergo microgametogenesis, that we are going to learn. Microgametogenesis to develop the pollen grain, male gametophyte. And yes, in the next page, which I am going to explain you about microsporogenesis and microgametogenesis. I hope you got the structure of microsporangium with this diagram, and you please practice this diagram. Question can be asked, draw a label diagram of internal structure of microsporangium. Yes, I hope you got it. The very importance of each layer, that epithelium is protective. It is a protective layer, and these 3 layer, that including 1, 2 and these 3 third, that they also help in dehiscence. They also help in dehiscence of pollen grain. Help in dehiscence of anther, so that to release the pollen grain. An innermost layer, this is a nursing tissue. This is a nursing tissue that nourishes the developing microspore and pollen grain so as to form male gametophyte called pollen grain. And yes, in the next video you will learn what is microsporogenesis and what is microgametogenesis to have the full development of male gametophyte. Now, on next page, let us continue with the 2 processes, microsporogenesis. Yes, microspore mother cell is diploid. All microspore mother cell which present in that microsporangium, they are diploid. They undergo microsporogenesis. What is this process? This process is occur through 2 steps. These 2 steps are meiosis 1 and meiosis 2. What is meiosis 1? Is a reduction division, and this is equational division that you learn. Equational division. Here this undergo reduction division, produce 2 haploid cells. Each haploid cell undergo meiosis 2 and produces 4 haploid cells. These are microspore. What these are? Microspores. What is this process? Process of production of microspore from microspore mother cell. So the process of production of microspore from microspore mother cell is known as microsporogenesis, and it occurs through meiosis 1 and meiosis 2, producing 4 microspore. Now, each microspore separately, they are together but separately under the process of microgametogenesis. So here I am going to write microgametogenesis. What is microgametogenesis? That each microspore undergo 3 successive mitotic division. Each microspore undergo mitosis, unequal mitosis division, unequal mitotic division. Now what is that unequal mitotic division? Suppose, let us take, because there is no place, we can start from here. This microspore undergo microgametogenesis. That what? It undergo microgametogenesis having this nucleus over here and vacuole. Now second step, there is a spindle formation. Toward one side having 2 large vacuole. Now this what spindle formation, it is to divide that nucleus into 2 nuclei and forming one large cell called vegetative cell and small cell called generative cells. These generative cells remain in swimming condition inside this vegetative cell. So here this is called 2 nuclear stage. It happened all 4 microspore, that is microgametogenesis. Internal layer is called as intine, is made up of cellulose and pectin, while that outer layer is called exine. It is made up of sporopollenin. It's a special protein, most resistant protein, can be asked in NEET. One of the following is the most resistant protein, most resistant protein. And because of this most resistant protein, this pollen grain can found in the form of fossil, and therefore pollen grain can found in the form of fossils. And remember, for NEET, that assertion and reason question can be asked. Pollen grain found in the form of fossil is assertion. Reason, exine is made up of sporopollen. Okay, this is a 2-cell stage. 60% of the pollen grain they shade in 2-cell stage. This is to be remember. 60% of the pollen grain they shade in 2-cell stage, and remaining that 40% that shade in 3-cell stage. What is the 3-cell stage? It's supposed to draw here. See now, here this is a vegetative cell with this generative nucleus which divided into 2, and this is a vegetative cell. This is covered by intine and outer covering is exine as usual. So once again, to label: exine, intine, this is generative nucleus or generative cell with 2 male nuclei and a vegetative cell. This is called male gametophyte. Each one is called male gametophyte. also can be called as carrier of male gamete. So this is very important to note down. Now, as I told you that all these microspore simultaneously undergo microgametogenesis. That means all the four develop together and therefore they form a common structure. All together as they are together. See, these are the four microspore. These are the it can be, to understand better way, see, these are the 3 cell and that 4 cell is this one. That is, just imagine behind. So this is 1, 2, 3 and this is 4. Now as a common structure, so no doubt they have this, they have this entine, they have their own entine and they have their own exine also. So these structures 4, they are within with their complete development and commonly hold together. They are commonly hold together by a common structure called callose. This is a non-cellular structure. This is non-cellular layer that hold these 4 together called callose. So see, these 4 pollen grain, 4 male gametophyte hold together by callose. This structure is known as tetrad. Now, as in previous video, we learned that anther has these 3 layers: peri-epithelium, endothecium and middle layer. They get ruptured. They help in dehiscence of anther to release the pollen grain. So, in this, at maturity, this wall, longitudinal wall, get split because of these 3 layers and pollen grain release separately. As they are initially in tetrad stage, but later they become free and they release. That is what pollination, transfer of pollen grain from anther to the stigma, that is pollination occur either in 2 cell stage. As I told you, 60% of the pollen grain they shed in 2 cell stage, or 3 cell stage having vegetative cell with 2 nuclei. Now, very important is that this is a carrier of male gamete. Pollen grain is not a male gamete. These 2 nuclei are male gamete. This entire structure is called male gametophyte. Thus, the process is development of male gametophyte through microsporogenesis and megagametogenesis that we learn. As this is mentioned, you can refer on NEET also. These diagrams are given on NEET that you can go through. This is the anther where you can find that how pollen grains are developed. These pollen grains are developed. This is a one microspore. As I told you, these are the 4 microsporangium present in each anther. Each microsporangia has porogenous tissue. Porogenous tissue has that microspore mother cell which undergo meiosis 1 and meiosis 2, producing 4 microspore. And these 4 microspore undergo microgametogenesis, that is development of male gametophyte, where these pollen grains are developed through the process of microgametogenesis, that is unequal mitotic division, which already I mentioned over here. This is unequal mitotic division because one large cell and one small cell is produced. And finally, this 2 cell stage which is produced, that shade in the form of, this is what 2 cell stage, that shade 60% of pollen grain. That means pollination occur all on the stigma and 40% around they shade in the form of 3 cell stage. And further process is, that is development of pollen tube through pollen nuclei or tube nuclei. This tube nuclei that develop the pollen tube carrying these 2 male gametes, one fused with egg cell, another fused with polar nuclei to form zygote and endosperm mother cell, and that is what process and part of the reproduction. I hope you did understand what is the process of development of male gametophyte through this explanation. This first slide was study of internal structure of microsporangium or anatomy of microsporangium, where question can be asked: nursing tissue tapetum based on function of middle layer, endothecium and epithelial layer. That question can be asked, as well as arrangement of these 3 layers inside to outside and outside to inside also can be asked in NEET. That one of the following is a correct sequence of presence of these 4 layers around microsporangium is from outside epithelium, endothecium, middle layer and tapetum. Which one of these is nursing tissue? Tapetum. So these tapetum, especially to explain about tapetum is very important, as tapetum also consists of Ubi's bodies. This Ubi's body produces pollen kit, and this pollen kit, that is what yellow color, yellow viscous powder that help pollen grain to stick on the stigma, as well as tapetum produces and release enzyme, release calcium and other mineral, as well as carbohydrate, fat and protein and nutrients. So these nutrients along with calcium and enzyme help to nourish the developing pollen grain or male gametophyte. Therefore, this tapetum is also known as a nursing tissue. Please remember, as in human being, in male Sertoli cells, they also provide nutrition to developing sperm. Therefore, in human being male, that Sertoli cell is a nursing tissue, and in plant, tapetum is a nursing tissue. That both this tapetum and Sertoli cell help in the nursing of male gametophyte, okay, or male gamete. I hope you did understand that anatomy of microsporangium and development of male gametophyte. And I ask you to please practice this diagram. Question can be asked for 3 mark or 5 mark with diagram. Explain process of development of male gametophyte. And in NEET, question can be asked on this main 2 point, that tetrad is held by a special non-cellular material ABCD, callose, and pollen grain found in the form of fossil because of most resistant protein that form exine ABCD sporopollen. I hope you did understand entire process of development of male gametophyte. Even if you are in doubt, you can ask in comment section. I appeal you to subscribe our channel as well as like it and press bell icon so as to receive new videos as and when they are uploaded. Thank you.